In this laboratory, we're going to look at nervous tissue and neurons. I've shown you this image before of a multipolar neuron. This neuron comes from the anterior horn of the spinal cord. I wouldn't expect you to know that at this point in time clearly, but you don't have any trouble identifying the nucleus and the nucleolus and the cytoplasm, which is called the perikaryon in neurons. It's the cytoplasm within the cell body of a neuron. The cytoplasmic Nissl substance, which is all this granular or punctate staining, essentially consists of rough endoplasmic reticulum and polyribosomes. This stains basophilic, and it occurs in clumps because what you're going to learn later in the course is that the substantial microtubule networks disrupt the continuous sheets of the rough endoplasmic reticulum, so that when a neuron cell body is stained, you see these clumps of material, and this is given the name Nissl substance. You can observe dendrites that are coming into this neuron cell body, and can maybe just barely see the axon hillock in this area. You'll notice that the Nissl substance is in the dendrites, so there is rough ER and ribosomes in the dendrite, but the axon hillock appears unstained, and that's because there is no rough ER or ribosomes to speak of going down into the axon of the neurons. This particular neuron is stained with toluidine blue. I don't really expect you to know that. And then you can see some various nuclei of supporting cells around this multipolar neuron. Here are some more multipolar neurons that are stained with the silver stain. Neurons preferentially take up heavy metal, and you can notice a lot of neurofibrils on this slide. These are essentially aggregates of neurofilaments and microtubules, and these filaments run throughout the cytoplasm in neurons. Here are some more multipolar neurons that are silver stained, and again, these are in the anterior horn of the spinal cord. You wouldn't be expected to know that at this point in time, but you can see this network of neuropil or cytoplasmic processes from these various neurons. Here are several peripheral nerves. This section demonstrates two peripheral nerves actually in cross-section within dense irregular connective tissue. You can identify axons within these nerves, and you can identify flattened cells of the perineurium. Nuclei associated with the axons could be either nuclei of Schwann cells or cells that make up the endoneurium. And we'll talk about the endoneurium and perineurium later in the course. Here are some more peripheral nerves. This is a section that's stained with osmium, and you can see myelinated axons, and you can see the perineurium like this, and the epineurium like this. These dark staining cells are actually fat cells, or adipocytes. The endoneurium, which are connective tissue sheets that are around individual axons, cannot really be delineated at this low magnification. Here's another peripheral nerve. This time it's stained with HNE, and you can identify individual myelinated axons. You can also see nuclei that could either be Schwann cells or fibroblasts, and at this magnification you can't really distinguish between the Schwann cells or fibroblasts. So here is a smaller nerve and a little bit larger nerve. Now something I'd like to highlight for you right here where my pointer is, that's the actual axon that's being wrapped with myelin. Here is a peripheral nerve with some surrounding connective tissue, a lot of fat shown here, some small blood vessels, maybe an artery and a small vein here, and then a larger artery shown here. And in this larger artery, you can clearly identify something called the tunica intima, the internal elastic lamina, like so, the tunica media, which is mostly smooth muscle, and then the tunica adventitia. And you're going to come to appreciate that the tunica adventitia in these arteries blends with the loose connective tissue around the nerve. And you're going to, I think, remember from gross anatomy, oftentimes that blood vessels and nerves run together, and that's what you're observing in this particular histological preparation. This is a little bit higher power view in longitudinal section of myelinated axons. You can see a nice myelinated axon running like this. The dark arrow is highlighting a node of Ranvier, and you can see the light blue staining connective tissue. This is a trichrome stained slide, and these trichrome stained slides, the blue stain stains connective tissue. This would be the endoneurium that's enveloping each individual myelinated axon. So let me review this for you one more time. This would be the axon itself. The white area would be the myelin sheath, which is kind of dropped out in the histological preparation, the node of Ranvier like so, and then the connective tissue that would make up the endoneurium. Here you can see some nuclei, and these nuclei may be a nuclei of Schwann cells or fibroblasts. Of course, the Schwann cells are producing the myelin that wraps around the axon, and the fibroblasts would be the connective tissue cells that are making the endoneurium. This is a plastic section stained with toluidine blue. You can observe the node of Ranvier at the dark arrow head, and you can also identify the nucleus of a Schwann cell, probably visible here in the upper left portion of the field. In this particular preparation, the myelin stains a very deep blue, and the axons appear unstained. This is a low-power view of a spinal ganglion. In this micrograph, you can clearly see the dorsal root towards the top of the field and the ventral root on the bottom of the field, and perhaps the spinal nerve over here on the right side of the field. A higher power view of the spinal ganglion, you can identify nerve cell bodies. These big cells are the nerve cell bodies. Maybe right here would be part of the axon hillock, and some of these nerve cell bodies you can get a sense of the nucleus and nucleolus. You can see the punctated staining of the cytoplasmic Nissl substance, and you can see some smaller cells which would be supporting cells or satellite cells around these ganglion. This is a fairly high-power view of the cortex of the cerebellum, and you can clearly notice the white matter shown here and the gray matter that's shown here. And it's very easy to get your orientation in the cerebellum. If you look at the gray matter, there are several distinct layers. There's a granular layer, a layer of Purkinje cells shown here, and the molecular layer. And so you can always tell your orientation of gray matter from white matter in the cerebellum by looking for the Purkinje cell layer. And you're going to come to appreciate that the Purkinje cell layer is a layer of cells in the gray matter that's between the molecular layer and the granular layer. Here is just a higher power view of the Purkinje cells shown like this, so a little bit of the granular layer on the bottom and the molecular layer on the top. In some of the Purkinje cells, you can actually see the nuclei and the nucleoli. And you can get some ideas of profiles of small blood vessels in various positions on this slide. And a silver-stained preparation, just a higher power view again of the Purkinje cells, and you can see some of the cytoplasmic processes coming off of these Purkinje cells going into the molecular layer. This is a Golgi stained preparation of the cerebrum, and it's just to highlight these large pyramidal cells and various protoplasmic astrocytes. On the pyramidal cells, you can notice the very large apical dendrites. Just a thought question for you on these pyramidal cells. These are all the dendrites, so where might you expect the axon to be? Just some various profiles of protoplasmic astrocytes, perhaps a little bit of a profile of a blood vessel on the bottom. This is located in gray matter, and this is a silver stain. Another view of protoplasmic astrocytes. Protoplasmic astrocytes are actually the largest of the glial cells, and they tend to possess relatively blunt cytoplasmic processes. I'm not going to expect you to really differentiate between protoplasmic and fibrous astrocytes. You can make a general rule, though. Protoplasmic astrocytes are found in gray matter, whereas fibrous astrocytes, which are cells like this, are found in white matter. Fibrous astrocytes tend to have very very fine filamentous cytoplasmic processes. This is not a course in neurobiology, so I'm not going to ask you to really be able to distinguish protoplasmic versus fibrous astrocytes. As you become more experienced, you'll be able to identify areas in the central nervous system that are gray matter, because those are the areas where you find the cell bodies of neurons, as opposed to the white matter where you essentially are just finding the axons and no cell bodies. Here are some views of oligodendrocytes. Oligodendrocytes are small glial cells, and they're usually found near nerve cell bodies, and we believe that the oligodendrocytes function like Schwann cells in that they myelinate axons within the central nervous system. So for example, here's an oligodendrocyte. Here are some individual cell bodies of neurons. So here, some more oligodendrocytes. And you can get the impression that these oligodendrocytes are putting out cytoplasmic processes that are wrapping around individual neurons. And one thing that's rather interesting, within the central nervous system, these oligodendrocytes may envelop multiple neurons with myelin sheaths. These are small microglial cells. These are thought to be the macrophages within the central nervous system. Here are some pigmented neurons. These neurons are actually pigmented with melanin granules. These happen to be within the locus ceruleus of the pons. Pigmented neurons are relatively rare in the central nervous system, but they are found in the olfactory bulb, the substantia nigra, maybe reticular formation, and they're found in some spinal and sympathetic ganglion. This particular cell is just meant to demonstrate the pigmented neurons. I'm not going to ask you to locate what specific region in the central nervous system these happen to be found within, but you can clearly see the neurons with melanin granules within them. Here are some blood vessels that you can see in this region of the central nervous system. Here is a Meissner's corpuscle. Meissner's corpuscles are touch receptors that are located in the hairless regions of the skin. You find them within interdigitations between the dermis, which is down here, and the epidermis here. And I'll remind you, since this is skin, this is a stratified squamous epithelium with keratin. The dermis is a irregular connective tissue. You can see the Meissner's corpuscle that I'm highlighting here. These touch receptors consist of axons, perhaps of 1 to 4 different types of afferent nerves that are not visible, but these nerves are surrounded by an elaborate club-shaped capsule which is composed of fibroblasts and flat epithelial-like cells. And nuclei of these cells tend to be located horizontally. The axons, once they enter this capsule, lose the myelin sheath, and they're going to branch extensively and interdigitate processes among the epithelial-like cells. And this is just a little bit higher magnification of the Meissner's corpuscles. And finally, this onion-like structure is a Pacinian corpuscle. This is a pressure receptor, and in this case it's found at the junction between the dermis and the hypodermis. And so here's the hypodermis, the fat, and more of the connective tissue in the dermis. This Pacinian corpuscle has multiple layers of concentrically arranged fibroblast-like cells that comprise the capsule of this structure. In real life, these concentric layers of cells are filled with fluid. Right in the very middle, you can actually see the axon for this receptor. You can see these nuclei. They would be nuclei of fibroblasts, which are the cells that are making up these concentric rings of tissues that are surrounding this axon. You can see some small blood vessels off to the side in this image as well.